Manoranjani as well. If you really don't want your videos to be on, you can give an excuse of poor network connection.
No, sir, I will be keeping my video on
Not
during the
you, the
presentation.
others.
Okay, sir.
Sir, Mano, you are on live and you can start the session.
Okay.
Kirtan?
Can I start, sir?
Yeah.
Okay. Good evening, everyone. A very warm welcome to all the respected dignitaries and Dr. Guhan Ramamurthy, sir, state leaders and dear medical students who have joined us today. It gives me immense pleasure to welcome you all to this academic session organized under RIT 2026. I am Kirtan, a second year MBBS student from Tagore Medical College, and it's a privilege for me to serve as a host for today's session on behalf of the RIT subcommittee of IMA-MSN, Tamil Nadu State Branch. Then we have our co-host Mugia.
Hi everyone, I am Mugia, final year MBBS student from Government Medical College, Tiruvallur, and I am also a host along with Keerthan.
Then, on behalf of the IMA-MSN and Junior Doctors Network, Tamil Nadu State Branch 2026 to 27, I extend a warm welcome to all the respected dignitaries and all the participants who have joined us today for this journal presentation session. At the beginning, I would like to respectfully acknowledge the constant guidance and encouragement provided by the patrons of the IMA Tamil Nadu State Branch. Dr. B. Sridhar Sir, State President IMA TNSB, Dr. B. Sengutuvan Sir, IMA Past President IMA TNSB, Dr. A.K. Ravikumar Sir, President Elect 2026 IMA TNSB, Dr. N.R. Tyagarajan Sir, President Elect 2027 IMA TNSB, Dr. N.M. Thiraviyam Mohan Sir, State General Secretary IMA TNSB, Dr. D. Solomon Jaya Sir, State Finance Secretary IMA TNSB. I would also like to convey my sincere regards to our state leadership team, Dr. R. Annamalai Sir, Chairman IMA MSN TNSB, Dr. S. Jamunarani Ma'am, Co-Chairman IMA MSN TNSB, Dr. V. S. Hariharan Sir, Chairman IMA JDN TNSB, Dr. Kannan Rajendran Sir, Convener IMA MSN Subcommittee, and a special appreciation to the state team members who continuously support the activities of the Medical Students Network. Arunagiri Sir, Convener IMA MSN TNSB, Ajay Karthik Sir, General Secretary IMA MSN TNSB, Bala Hariharan Sir, Co-Convener IMA MSN TNSB, Hariram Sir, Co-Convener IMA MSN TNSB. So, RIT 2026 is a dedicated research initiative functioning as a subcommittee with the vision to strengthen research knowledge, critical thinking, and scientific approach among medical students. Through this platform, we aim to nurture curiosity and promote evidence-based learning. Today marks a special milestone as this journal presentation is the very first initiative of RIT for this year, and we are truly excited to begin this journey with all of you. And today's topic is Engram persistence, unraveling memory retention in retrograde amnesia. So we have a short introduction for our Dr. Guhan Ramamurthy sir. He is a distinguished neurologist with strong academic credentials and international recognition, including the prestigious Fellow of the European Board of Neurology, FEBN. He has undergone advanced training in neurorehabilitation at the National University Hospital, Singapore, reflecting his expertise in managing complex neurological conditions and promoting functional recovery. With dual super specialty qualifications in neurology, DM and DRNB, Sir has demonstrated exceptional dedication to both clinical excellence and academic advancement. His work emphasizes evidence-based practice, patient-centered care, and continuous learning. In addition to his clinical contributions, he is deeply involved in teaching and mentoring medical students, helping bridge the gap between theoretical knowledge and real-world neurological practice. His areas of interest include cognitive neurology, memory mechanisms, and neuroplasticity, making him exceptionally suited to moderate today's session. We are truly honored to have you with us today, sir. I now request Dr. Guhan Ramamurthy, sir, to kindly share a few opening insights and guide us into this session.
Okay, thank you for the wonderful introduction, Kirtan, and it's nice to be part of the young budding medicos. And it's a great initiative that you all have here as a part of research, because understanding research at this point in time is very crucial. And I actually have gone through the slides of Manoranjani, and I felt that to understand PICO questionnaire at this point in your MBBS, wherein you're all stuck between your books and looking forward to NEET exam, and at this point in time looking into research is of very important. because the moment you are into research, you're looking at scope beyond the NEET, because every entrance examination that lies abroad, including your US, PLAB, and everything, they are going to look into the research that you have done during your undergrad day. And doing a research is not important. Not just doing a research is important, but understanding research is most important. Why are you doing a research on the first place? What is the question that you got to answer? That's very important. And Manoranjani, because you chose the topic of hippocampus and amygdala, which is a beautiful neuroscience topic that we can have a wonderful discussion. But let me put forth few questions before you proceed. So upfront, the next thing is going to be the presentation by Manoranjani, right? Or
Yes,
is
sir.
there any
Yes.
other formality before that?
No, sir.
Okay, so Manoranjani, I just don't want it to be a regular presentation, because the moment you start that, I'm not sure how many would know about the memory engrams, and how many of you here don't know memory engrams. Put up your hand, not text message, but put up your hand. Okay, so whoever is not going to put up their hand, I'm going to ask them questions, including Surya Prakash, Archana, Akash, Gokul. So I assume that they know about memory engrams. So, or either they are not awake. So this is the reason that if you go into the memory engrams right now, they might not understand the whole paper. So I want you to explain the basis of why you took this paper, the basic neuroscience of the paper before going into the slides, because the moment you go into the slides, it's altogether a different scenario. So I wanted to explain the basics of whatever you understood in the basics of neuroscience of the memory. Please go ahead. Your mic is muted.
Am I audible?
Yeah.
Yes. Good evening, everyone. I'm Manjini Meditation, audio and distributed in family course.
Your voice is breaking actually. The others can also put down their hand.
Okay, sir. I'm audible now.
Still, it's breaking. Is it for me alone or is it for others as well? Kirtan, do
So
you
you
hear?
can turn
Yes.
off your video so
It
that
is,
it will not
it is
break.
breaking, sir.
Okay.
Okay, I hope
Or you
it's
can turn
clear
off your
now.
video and talk.
Yeah, this is now better.
Okay. So the main reason for me to take this paper, why? A research is basically always starting with a question in our mind, right? So here came a question to me that what amnesia is basically. We all think that it's memory loss, and we might think that it is okay. Once the memory of past memory is lost, it is inaccessible. Now here, this question came into my mind. Okay, whether it's exactly memory loss or is the memory not accessible? Whereas like any cells, specific cells, anything else, like instead of normal synaptic neurons, can we get the memory back? Then here comes the rescue of engram cells. So that was the main reason for me to take this paper into consideration.
Okay, that's wonderful. It's a great initiative to take up this paper specifically. And so let me put forth to you further questions, because unless you're strong with this, I'm not sure you would really understand the essence of this paper, because not only the framework of the research, but also the basics of neuroscience is very important before you delve into the depths. And this paper does not give you the basics. So I wanted to know if you went further. So tell me, what are the types of memory that you know of, and how are memories formed on the first place?
Yes, sir. Am I audible?
Yeah, very much.
Okay. So there are different types of memory. As first basic we know is the short-term memory and the long-term memory. A short-term memory that lasts from seconds to minutes, whereas a long-term memory it goes from hours to lifetime, which is formed after consolidation. Memory consolidation is a step where the short-term memory is converted into a long-term memory. Apart from that, we also have something called as sensory memory. which last like milliseconds to seconds. Like when say any immediate sensory input via vision or sound, we like seeing a flash of light, that kind of memory we can think is a sensory memory. And based on the concept of how it is formed, hello,
Yes, yes, very much. You're on the right track.
okay, sir, okay. First, whenever memories come, I'll try to explain in my own words. Encoding. It means the brain immediately starts to get the inputs of the particular information, whether it's visual memory or any sound, whatever it is. Second, it reaches the information side. This information is retained, whether in temporary or in permanent side. The third step will be the stabilization of the memory, that is our memory consolidation, which might occur over hours to days, and this requires protein synthesis. Now, this happens in our brain hippocampus. Any two neurons.
Voice is actually a little muffled. We can hear it, but the words are not clear between each other.
Okay, sir. I'll try to.
Yeah, maybe the mic is too close to the mouth.
Now it's clear.
Yeah.
Yes, sir. Sorry for the network issues. Okay. So first we said was encoding, the input of information. The brain starts to encode, get information from any visual or sound through whatever it is, some from sensory. Second will be the storage, where the information is retained either temporary or very important crucial step, that is memory stabilization. This is done by the memory technique called memory consolidation. Memory consolidation is where you simply convert a short-term memory into long-term memory, and we need protein synthesis for this. You need to connect the two neurons to any. You have to connect a small short-term memory to long-term means the connection between two neurons should be strong. So we need protein for them. That last will be the retrieval, memory retrieval, recalling the stored memory.
Okay, that's
Yes.
great. Have you completed, or do you have any more to say?
Uh, this, this much, that's it, sir, from my side.
Okay. So, what is episodic memory? What is procedural memory? And what is semantic memory?
Episodic memory.
Yeah. Anytime you want, you are free to say the question. You are free to pass the question. So don't worry about if you don't know the answer.
I will try my best, sir. Episodic memory, it can be from any experiences, sir, our own personal experience, from time or any place. If
Wonderful.
I'm
Eh,
right, any
Yeah,
incident in the past.
it's an episode, whatever the incident that has happened yesterday while I was having my breakfast. That is the episodic memory. What is the semantic memory?
Semantic. Uh, uh, I'm not very sure about it, sir.
Okay. And what's the third one? That's procedural memory. What is a procedural memory?
Procedural memory.
Yeah.
Okay. Is it like a memory, kind of an implicit kind of memory, sir? Any skill or any motor activities that will be like muscle memory, for example? Can we take?
So not exactly muscle memory alone, but it's a procedure that like driving, like cooking. These
Ah,
how do I cook a sandwich? So the knowing about this sequence of cooking a sandwich is a procedure. Driving is a procedure. So this is procedural memory. Semantic memory is describing things. It's the semantics of a particular thing. How does my phone look like? It's a black phone, it's long, it's 4.5 inches screen, and it has 1,000 lumens in brightness. These are the descriptions that I'm telling from my memory, but it is not about an episode, it's about an object. So describing the object is a semantic memory. And episode is a common thing that we talk about. Like what happened yesterday, what happened day before yesterday, what happened while I was doing my undergraduation. This is episodic memory. And what is procedural memory and episodic memory and semantic memory? What have they got to? Why do I need to know about these things? Is episodic memory is the one that takes place in the hippocampus. The hippocampus, the medial temporal lobe, whatever you're talking about, the memory consolidation, everything is the episodic memory. The semantic memory description about objects is in the anterior temporal lobe. If it is a verbal semantic, it is the left anterior temporal lobe. If it is a visual semantic, it is the right anterior temporal lobe. The procedural memory, because it is concerned with motor activities, it it is localized in basal ganglia and cerebellum. So the more you get to do things, that's basal ganglia and the cerebellum, and they store the procedure. So these are the three things. The one you told about short term and long term memory is with the due course of the time, whatever you were telling with. And specifically the short term and long term, we attribute it to episodic memory. The episodic memory, did this episode happen today or did this happen long time ago? The reason being, a short term memory, if it is very short, like the sensory one that you were talking about, few seconds, it's in the attention network. It's the dorso-parietal attention network. It does not go beyond this. If it is few minutes to hours, that's when it goes into the hippocampus. And then when it goes beyond days to months to years, that's when it goes into the long-term memory. So this is the categorization of the memory. But now, since we are concerned about hippocampus, you understand this, right? The short term, long term, this is with due to the time, and the type of memory is episodic, semantic, and procedural. This is to do with where the memory is stored for this. Since we are concerned about episodic memory, let's come to the episodic memory in the hippocampus. So let me ask you the question again. What is this perforant pathway that you're talking about in the hippocampus? Can you explain it a little further? Manoranjani?
Hello? Yes. Am I audible?
Yeah.
Yes, sir. So the Papin circuit is what it trains the memory circuit of the brain, if I'm right.
Mhm. Okay. Can you explain more about this paper? If anyone does not understand any point now, please raise your hand, because the moment you don't understand this, you won't be able to understand the paper. And if you don't understand the paper, you wouldn't understand the basis of the research at all. Everyone understands? Those who understand, please raise your hand. There are chronic non-hand raisers. Okay, that's great. Okay, so you can proceed with what is this purpose circuit?
The Papin circuit.
Yeah.
Yes, sir. Basically, this circuit is a neural pathway in our limbic system, which is involved in typical memory and emotional processing. So this has a particular sequence. Starts at the hippocampus, then it goes to the fornix in our brain. We are there. We have a body called the mammillary bodies, which is present in the hypothalamus. So those signals travel through the mammillary bodies. Followed by that, the third area is the anterior nucleus of the thalamus. Over there, they come through and then they pass through cingulate gyrus in our brain. And they come back to our hippocampus. What is the main role of this Papin circuit is memory. They link between the emotion and memory together. So
Okay, so
that is
what is the emotional part of the brain that concerns with memory?
emotional path.
The emotional. So with regards to the Papa circuit, it's the hippocampus, then it goes to the fimbriae, it goes to the fornix, through the fornix,
Yes.
it goes to the mammillary body, then through the mammillothalamic tract, it comes to the anterior nucleus of the thalamus, then to the cingulate gyrus, and then comes back to the hippocampus.
Yes,
So
sir.
now what you're going to explain further is within the hippocampus, about the perforant pathway.
Yes, sir. Emotional.
Let's come to emotion a little later, because we'll complete this now. regarding the perforant pathway that your whole research is all about.
Yes, sir. Yes, sir. Yes, sir. I can get you. A perforant pathway, if we put simple terms, it is an input pathway in the hippocampus where it carries the information from the entorhinal cortex to the dentate gyrus, if I'm right. Simply, from the entorhinal cortex to the dentate gyrus, they carry up. These, why we call it perforant? Because these fibers they perforate, they pierce through the particular hippocampal layers to
What
reach
do they
that
perforate
particular
through?
to reach that dentate gyrus. That's why we call them as perforant pathway.
What do
And
they perforate through?
the, via the dentate gyrus, sir,
Yeah, the perforant pathway, it perforates through what substance?
mossy fibers of
That's the third part. We are still in the first part.
sure.
What substance does it perforate through? That's
Substance.
subiculum.
Okay, sir. Okay, noted.
And then it reaches the dentate gyrus. So what's this perforant pathway about?
They help in main entry into the hippocampus, initiate the
Okay.
memory formation processes.
Okay, and then where does it go from dentate gyrus?
Dentate, they go into the CA3, if I am right. CA3 is nothing but particular region in the hippocampus.
The expansion of CA is,
Cornu ammonis 3.
what does it mean?
It is a particular subregion in the
The literal
hippocampal
meaning
region.
of cornua ammonis.
Yes, sir. It is a particular subregion in the hippocampus that helps in memory encoding and pattern of completion, if I'm right.
Okay. So where does it go from CA3 to? I mean CA3 TO2, where does it go to?
From CA3, they will go to CA1 pathway in the hippocampus.
Okay, where does it,
CA1 is nothing but the cornu ammonis 1 in the hippocampal region.
okay, where does it go from CA1 to?
From CA1, they go to the subiculum, followed by that to the cortex.
Okay, where is this placed in Papa's circuit? What is this to do with the Papa's circuit? Your perforant pathway, your mossy fiber, Schaffer collateral, CA3, dentate gyrus, subiculum. Where is this all placed in the?
Papi circuit.
Where is this placed in Papa's circuit?
At the initial stage of hippocampus, sir, because from there only in Papi circuit we start, and they come back to again hippocampus. From hippocampus to fornix to mammillary body, back to hippocampus. So on that path.
Wonderful. So this was way above the grade of MBBS because we study about trisynaptic pathway in DM neurology. Maybe I think the NEET PG syllabus is getting tougher and tougher, and they're asking you questions about trisynaptic pathway. But let me share you a PDF that I have trisynaptic pathway, because if you don't see this PDF, you wouldn't understand this paper, and this is very important. You see this? Are you
Yes, sir.
able
Yes,
to
sir.
see this? Okay. So this is what this is. Understand this. This is very important, and have all your brains active, because this part, what I'm going to tell now, is how a memory is formed. Okay. So whatever happens is that when you have a visual input, it goes to the visual cortex, it goes to the visual association cortex. When you have a sensory input, it goes to the sensory cortex, it goes to the sensory association cortex. And when you have an auditory input, it goes through the auditory cortex and auditory association cortex. From all these association cortices, it goes to the temporoparietal occipital junction. That is the junction area for all stimuli together. From this temporoparietal occipital junction, it comes to this entorhinal cortex. It goes to various places. One place where it comes to is this entorhinal cortex, because that is the gateway, as you were telling, into the hippocampus. Once it comes to the entorhinal cortex, it is like a memory engram. But how is this engram formed? It is like elementary substances are coming into the entorhinal cortex. They are trying to make a scene out of it. Okay. But once you have it in the entorhinal cortex, this episode that I am experiencing currently, if it is not getting engraved into this trisynaptic pathway, I wouldn't remember this. So, for a memory to be formed, you need to be attentive. If you don't have the attention, this is not going to go into the association cortexes. It is not going to come into the entorhinal cortex. Once you have an attention, once the input goes to the association cortexes, TPOG, and then to the entorhinal cortex, from there, it passes through this perforant pathway. The name perforant pathway because it pierces through the subiculum and comes to the dentate gyrus. From the dentate gyrus, it goes to the CA3, which is cornua ammonis, literally meaning sheep's horn, because it resembles the sheep's horn. That is CA3 layer of the hippocampus. From CA3 layer of the hippocampus, it goes to the CA1 layer of the hippocampus, and from there it comes back to the entorhinal cortex, completing a trisynaptic loop. So what do they actually do is that the entorhinal cortex, if it does not go to the dentate gyrus, no memory is formed. This dentate gyrus is where when you get an episode of memory, what does it say is that have I experienced this before? If I have not experienced this, then it forms as a new memory. If it is a has if I have experienced it before, it makes it as an associated memory. Once it gives a tag that it's a new memory, it goes to the CA3. From the CA3, it starts to make puzzles of all the events. What is the sound? What is the sensory feel? What is my emotion? Everything comes together into one plate. It's like a jigsaw puzzle. If you activate one area in this, then you feel the whole emotion. Say, for example, you had been to a Starbucks yesterday. You love the whole feel where you were with your very close friends. You had a very nice time together. Next morning, if you feel that coffee smell, the whole episode of the yesterday gets triggered. And this is because of this association in the pyramidal cells. And from there, it goes to the CA1. This is again where the actual should I have a new memory or is it an old memory? It is stronger in CA1 than in dentate gyrus. So this is how it is formed in pieces. And finally, is it a new memory? Then it goes through the entorhinal cortex and it forms a trisynaptic loop. Once you have studied something, once you have had a new experience, this information goes through the loop again and again and again and again. And the more it goes through, it is going to form new dendritic sproutings, new dendritic spines, because there is long term potentiation that's happening as the electrical impulses pass through this loop again and again and again. And this dendritic spine can be measured. The long term potentiation can be measured. What is the neurotransmitter for telling something is potentiated? It is AMPA. It is NMDA. It is a glutamate based neurotransmitter. If when measured is positive, it means there is a long term potentiation. This is the functional measurement. How do you measure structurally? It is the dendrites. The number of dendritic spine shows that there has been a long term potentiation. There has been an increased utilization of the pathway. So these are the two ways that you would actually strengthen the loop and strengthen the memory. When does this actually happen? It happens while you are actually sleeping. This is the reason why when you study something new, if you don't have a good sleep that night, there is no processing that's going to happen. And the processing would happen more in the REM sleep. And when does the REM sleep happen? It is the latter part of your sleep. So if you have 8 hours, The first 4 hours, there is very little REM sleep. The latter 4 hours, there is more REM sleep. So when do you think is you're gonna have more consolidation? The latter part of the sleep. This is why if you're going to sleep for 4 hours, your memory is not going to get consolidated. So whatever you read the previous day will not stick to your memory for a very long time unless you have had a very strong emotion attached to it. This is the reason why on the day of exam, before the day of exam, when you don't have a proper sleep, you still study, you don't sleep, your memory is not consolidated, but you are still able to remember the next day if it is the day before your exam, because there is very strong emotion of fear that is getting tagged to the memory, and that will have a very short lasting effect until you don't have another sleep to get it more consolidated. So this consolidation, once it happens over and over again, once it comes to the entorhinal cortex, for long-term memory, it goes to the respective association cortices. It sends to the TPOJ, the temporoparietal occipital junction. It sends to all association cortices, and from there it gets engrammed there. So there is a memory engram that's happening here in the trisynaptic loop. Engram meaning what are the facts of that particular scene, and is it associated with vision, is it associated with sensory, is it associated with auditory, is it associated with touch, is it associated with any emotion? All these features of a particular memory is what we call an engram. Okay, so how is limbic system associated with it? Let's see after the talk. I think this should be sufficient enough for you to understand the paper. Does anyone here have any doubts at this point in time? You can ask me, I'll explain it further. If no doubts, we'll proceed with the presentation of what Manoranjani has to say.
Yes, sir.
Any doubts, any questions, or you fully understood?
Why do we need a CA1 if already the dentate is perform the function of identifying the which is this is a new memory or it is an old one?
So dentate gyrus is not very strong in doing that. The actual role of identifying the memory, if it is an old one, is CA1. That's where your actual engram is. This is just an entry point. So the entry, so if you feel deja vu that I have been here when you have actually not been here, it is like wrongly flagging that memory in CA1. This dentate gyrus is just an entry point where it says that, okay, I'll have to form a new memory. There is high chance that it might be wrong that it's a new memory. It can be wrong, it's an old memory. The sensitivity of that dentate gyrus is very poor. But it's a very important junction. It is the only entry point into the hippocampus for it to form a memory. If you're asked one question as to which compares with the old memory to form a new memory, you say just CA1. If this point of dentate gyrus is confusing you, forget that dentate gyrus is even involved in comparing with the old memory, because that's the primary function of CA1.
Okay,
Okay.
sir. Thank you, sir.
Any other questions? Because this initial perforant pathway, that is like the entry point. You don't get entry into that, you're not going to form a memory. And every memory has a tag to it: fear, vision, sensory, and what do I do if such a thing happens? So that's with regards to the next fear conditioning that happens with amygdala. So we will see that after the presentation. If no
Okay, sir.
questions, then we can proceed with the Manoranjani's presentation.
Now, moving on to the core of today's session, it's my pleasure to introduce our presenter, Ms. Manoranjani Venkatesan, a third-year MBBS student from Government Medical College and Hospital, Tiruvallur. She is also the RIT Subcommittee Head, IMA-MSN TNSP, and has been actively contributing towards promoting research culture among students. Manoranjani, the stage is now yours. You may begin your presentation.
Thank you, Kirtan. Thank you for the introduction. I am going to present my screen now. I request Avinash to give access to the screen presentation.
Avinash, share screen settings la போயிட்டு change பண்ணணும்ப்பா.
It's actually available.
Yeah. Okay. Okay. Is
You
my
can.
screen visible?
Yeah, it's
Okay. Okay. I hope my audible is also clear.
audible. It's clear.
Okay. So hi guys, starting with my presentation. My topic is Engram persistence and revealing memory retention in retrograde amnesia. So let's start with the presentation. Today in this paper journal presentation, I'll be covering the introduction part about the paper, the research question in Pico format, evidence basis which consists of the background and existing literature, the study design, methods, result, discussion, the clinical context, and also finally the output. Now, as we always know, any research always starts with a question in our brain, right? So here is the question for us. Let's start with the clinical question. In our everyday practice, we do see patients with head trauma. We see patients with post-ictal states or any amnesic patients, right? So the fundamental question we have in our brain is the patient's memory is permanently lost or is it simply inaccessible? So with this question in our brain, we are going to look into this journal paper. Why this article? Why we have to go with this article? First thing, it's a first of its kind study. It means this is the first paper that is actually proved that has broken the old dogma that retrograde amnesia equals to destruction. Okay. Yeah. Researchers used a concept called as optogenetics engram technology here to prove that amnesic animals, here the animal we used is mice, to retain their memory at the cellular level. The relevance to practice. We all are medicos, right? So we might have seen patients in the hospital with amnesia, right? Amnesia, retrograde amnesia can come from any ways. It can be from any RTA cases, road traffic accident, or seizure, post-seizure cases, or Alzheimer's. This paper directly reframes how we conceptualize memory loss in these patients and raise a possibility that yes, with it is not completely memory loss, rather it is the access to that memory. How the possibility retrieval. So looking into this access, we might change the relevant therapy basis also to that rather than the synaptic restoration. Okay, this is about the paper. The title is Engram Cells Retain Memory Under Retrograde Amnesia. Author is Thomas J. Ryan. I've taken it from the journal Science. The main paper we can look into here is it's a first of its kind study and challenges the classical synaptic consolidation model. Before getting into the paper, before getting into the PICO format, I want to share a story with you guys. How this experiment is done. I would like to share it in a very story basis so that it will be easier for it to get it. Okay. Now, I hope my audible is clear, right?
Yeah, bro, it's clear.
Yes, okay, fine. This is a simple flow chart of what our researchers have done in this study. They are taking two mice. One is a normal mice, other one is a transgenic mice. Now, what is this transgenic mice? Basically, genetic mice is where you artificially you modify the DNA of a mice, right? So here we are importing something called as CFOS TTA CHR2 system. This mice will have this particular system inside it genetically. Now, what is this? It might collapse. What is this CCR? What is this CFOS? Simple. CFOS is nothing but an activity marker, which is going to get activated whenever a neuron is spiked up. Number two, what is TTA system? It is a genetic switch system. And number three, what is this CHR2? CHR2 is the main protein here, which is sensitive to the light, simply called as channel rhodopsin 2 protein. Okay. Now we are taking this two mice into account. Both these mice are kept in a box. Okay. Now they are given a mild shock. Now this shock will give a kind of fear to the mouse, which is going to be the memory that is stored inside the mice. Simple. Okay. After one, the mouse one, which is a normal mouse, is called as a control mouse, and the one we are going to genetically modify is the experimental mouse. The normal mouse is given, we are going to give saline, which is group 1. You can see in this green pathway, group 1 mouse where we are going to give the saline. And the group 2 mouse where we are going to give anisomycin. What is anisomycin? Simple, it is a protein inhibitor. Why protein? Why are we giving protein inhibitor? Very easy. First, whenever any memory consolidation need to happen, memory consolidation nothing but converting a short-term memory into a long-term memory. So for that, you need two neurons to bind very strongly. So in order to bind it very strongly, you need proteins. What does this anisomycin do? They go block those proteins so that memory is not completely stored. So it might get erased. That is what the concept here is. That's why they're giving anisomycin to this mice here. Okay. Now the anisomycin blocks the memory out. After 24 hours, both the mouse are kept in the same box. The mouse that is given with the saline and the mouse that is given with the anisomycin. Simple. The mouse are given with saline, it freezes up immediately. Whereas the mouse that is given anisomycin, experimental mode, it is not freezing up. Does anyone have any idea why it is not freezing up? Okay. Till now, was the concept clear? Somewhat?
Come on, guys, say something. We need you all to be active
Yeah,
because,
bro.
because
Yes.
even I don't think I could have explained this paper so beautifully. So you all either say yes, no to show that you're awake.
Okay.
Yes.
Okay. Thank you, Archana. Yeah. I'll proceed. Now both the mice are frozen up. The control mice, we have kept the both the mouse in the box, right? The control experimental is not freezing up. Now here comes the optogenetics. When we immediately, pass through the blue light, because the genetic mice, we have introduced a protein called as CHR2 protein. Na? That protein is a light sensitive protein. What does this protein does? Immediately when you pass a light to that mouse, you might when you immediately pass the light, these neurons get activated, which is nothing. Okay, I'm sorry. I'm okay. I'll keep it very simple. Once you pass the light, this CHR2, which is present in the engram cells, immediately get activated up, in turn, which is going to cause recalling of the memory. That is what the conclusion says here. So, as the end, what we can understand is that is the normal mice, which does not have this protein, right, CHR2, couldn't able to recall that memory. But the mice that were having CHR2, when the light was introduced to them, they were able to recall the memory. I hope I am clear somewhat. So further, you will try to understand a better way. Can I proceed?
Yes, sure.
Okay. This flow chart, it's a simple way, diagrammatic representation of whatever I've covered. As you can see very easily, box A, box B. Before giving this box A is normal natural recall method, and second one is where we introduce the blue light. And as I said, immediately when the blue light is introduced, the genetic mice which contains this CHR2 engram cells, they were able to recall their memory compared to the normal mice. Okay. Looking into our PICO format question basis of a paper. Who was studied? Okay, now you might understand what all terms are used here. The adult mice, adult male mice with transgenic mice, C4TTA, transgenic mice were studied. What are we studying here? Which region part of brain we are studying here? The hippocampal dentate gyrus part is studied here, right? Apart from that, we are also testing the CA1, CA3, which is nothing but a subregion part in the hippocampus, and the lateral amygdala engram cells also seen. There are no inclusion exclusion criteria is used. Intervention. As I said, what we are giving here to the mice, we are giving anisomycin. So simple injection of anisomycin is given immediately after the fear conditioning. The initial stage, if you remember, I said we are putting the mice in a box and you're giving a mild shock. The fear is going to be the memory, right? So immediately after the fear memory, we are giving this anisomycin. And the next step, as I said, the optogenetic stimulation of the Engram cells, which is done by the blue light. Easy. Third is the control. Control we use here is the, I said two mice, na? Control and experiment mice. Control is here the saline injection that we used, and non-Engram cells, M. Shelley cells we will consider here. No shock controls are here. The mice with Engram cells are labeled. No fear to confirm the fear specificity. Outcome. What is the expected outcome? The primary outcome here is whether the direct optogenetic engram activation restores that particular fear memory or not. We, at the end of this paper, we will able to understand this. And secondary outcomes are nothing but the AMPA and NMDA current ratios we will look into, and we might also derive the dendritic spine density also. Okay. Step 4. Evidence basis. What is already known? Simple. The protein synthesis inhibitors, anisomycin, blocks the memory consolidation. This is something we already know. And second, the late phase part, which requires protein synthesis, like the specific hippocampal neurons or anything during learning sensitivity. As I said, any memory consolidation, they require proteins. Third, blocking the, we might think blocking consolidation means memory is permanently lost, permanently destroyed. But here, this experiment breaks that particular theory. It's not permanently lost, rather, it is inaccessible. Coming back to the same point. Gap in the literature. Nobody had, no, we have always research paper does. Any research paper you need to do, you have to look into the gap in that particular paper. Literature. Nobody has directly examined what happens to engram cells during amnesia, at the time of amnesia, or whether bypassing will actually help in recovery of the memory. And second question is the question of storage failure versus retrieval failure. There's a difference between both. Whether we are not able to retrieve the memory or only the storage part is able to retrieve up. Easy. The context, the hypothesis is well founded, logically seen on engram cells. Right question with right time with right laboratory, the researchers have used. Apart from that, the contribution, the first proof that engram survives amnesia. That is what the first proof was given. Second, it also shows the synaptic strength and memory storages are separable. It proposes engram connectivity as a true storage substrate and also reframes the amnesia as retrieval failure, not erasure. Coming to this point. Next point. Okay. Study design. Okay, I'll throw a question, simple question to the audience. What are the types of research studies do you know? Yes, guys? Type of research studies, you know, cohort, case control,
Cross-section.
yes, cross-sectional,
RCT,
excellent, RCT, right.
meta-analysis, systemic
Super
review.
analysis. Yes, exactly. Now here we are not going to look into any of those studies. All we know is cross-sectional, KP studies, cohort, RCTs, meta-analysis. No, the researchers have done what type of study? It is called as controlled experimental preclinical animal study. It's a simple animal study. Now here the study population is our adult male mice, I mean C4TTA transgenic mice more specifically, not generable to humans directly. It is an animal clinical trial study. Blinding and masking, no explicit report is found over here, and potential for detection bias. Randomized method, animals are randomly allocated to the saline or the anisomycin groups. No formal concealments or stratification is done. Approximately 8 to 12 experiments they are proven to be done over here. And inclusion exclusion criteria, very easy. A successful viral injection, a genetic mice is considered, and a confirmed engram labeling by fluorescence is taken into inclusion. Exclusion, easy. Failed viral expression and insufficient, the protein, the CHR2 protein labeling is not there. We are going to exclude those mice. Simple. Bias assessment, the performance bias is done. Anisomycin, we know it causes nausea and stress. Reduction bias is also done, but blinding not formally confirmed over in this study. Confounding factor, stress response is seen from the anisomycin, which could affect this freezing behavior. Next, the methodology. Coming to the methods. Approach. The methods were rigorous and consistent over here. Same viral labeling, that is CFOS TTA, is used throughout all the same mices, and same fear conditioning protocol, this context B, is applied to all the animals. Easy. At the same time, with same optogenic parameters, with 473 nanometers and 20 hertz and 50 ms across groups. Multiple independent experiments replicated over the core finding. Follow-up is also done over here. 1 to 8 days of post-fear conditioning with follow-up of complete all the mice that of groups, and no dropout were reported over here. And longevity of memory rescue is also tested upon at the 8th day. Outcome. Here the researchers have proved the freezing percentage during the light on and light off epochs, and they've also given the AMPA and NMDA pathways current ratio with the dendritic spine density and engram connectivity, amygdala C4 activation, and outcomes appropriate and considered with the research question. Statistical consideration. Here, the main thing we have to consider sample size calculation. It is not reported in this study. Any study you do, you need a particular sample size calculation, is what we usually think. It's not reported. 8 to 12 mice were used in the experiments accordingly. Effect size reported? No. No specific effect size reported over here. Okay. Coming to the results, most awaited part. So the primary result is the amnesic mice showed full fear recovery, full fear memory recall when engram cells were directly stimulated with blue light. As I said, the initial story, how right, that came as a primary outcome for us, which made a huge, drastic change in the normal thinking uh, of our study. Like, we used to think memory is lost, it's not retrieved, right? But this study has broken that part. Amnesic mice showed full fear memory recall. Here, the effect size is the saline mouse up to 60% freezing is seen, and the anisomycin natural mouse recall 10% freezing is seen. Anisomycin induced recall mice 60% freezing is seen, according to the study. Secondary outcomes, as said before, the AMP and NMDA ratio is elevated in the saline mouses, but is absent in the anisomycin engram cells. The dendritic spine density is also noted higher in the saline engram cells, no change in the anisomycin groups. Apart from that, the CA3 connectivity, as said in the part of the hippocampus, the engram to engram connectivity were also preserved in both the groups. Statistical significance, its p-value is less than 0.05. Supplementary data, they have provided a certain subgroup supplement data where they have said the cyclohexamide is used over here with replicated findings, and also the anisomycin is given 24 hours post training. So no amnesia was confirms the consolidation window is what they have proved over here, and no freezing also seen on light stimulations. There is no shock control. So this graph just gives an overall idea of what is engram connectivity and what is synaptic strength means. Engram connectivity, simple, they are formed during encoding. They are protein synthesis independent, whereas the synaptic strength, they require protein. They require protein synthesis and they are lost in amnesia. The engram connectivity, it's simple. It depends on the CA3 circuit pathway that Sir has discussed initially. Here, the synaptic strength, they provide cues to access to the particular engram pathway. This bar graph right over here, it gives overall percentage of how much as a result. 62% are actually p-value is less than 0.5. Peer memory recall was seen in natural saline, whereas in anisomycin 10%. Compared to the light stimulation, 58% is seen in saline mice and 55% is seen in skewed anisomycin derived mice. Okay, coming to the discussion part, the strength and the limitation. Every paper, when we look, we have to look into both pros and cons, right? So here the strength. It is a multi-method convergence paper with all includes the behavior, the electrophysiology, the morphology, and also the circuit, tracing all the points to the same conclusion. Apart from that, the rigorous control. It pad cell recordings were used. No shock, as I said, second supplementary, that will come over here. No shock controls, dread negative controls, and two different PSIs are actually seen. These are the strength points we have to look into. And apart from that, replicated across brain regions are also being seen as a secondary outcome. Limitations, yes, as we know, it's an animal study. Mouse to human translation is speculative. The anisomycin is non-specific. It might cause stress and nausea, as I said before. Third, the blinding, which is not formally reported, so it might risk the detection bias. No power calculations. Specific sample size is not justified over here. The molecular basis of connectivity preservation is not identified, and all mice, well, like male mice, is what differences is not explored. That is also we have to consider here. Do results support conclusion? Yes. Here, the they have said 5 independent experimental approaches actually converge the same conclusion. Conflicts of interest, it's not declared explicitly, and they just who have funded this project they have given, like in Brain Science Institute and everyone. Yeah. Clinical context, clinical applicability. This is the most important part. Now, as we look, the patients cannot be directly applied. We cannot use this technique to directly to patients because the preclinical mouse study is, however, needs furthermore intervention. But the therapy, right? The therapy part, we might, change the therapy part. We might change our thinking that yes, Engram cells can be, activated, and based on that, we can give the therapy basis. Yeah. Change in practice that does not change the current clinical practice directly, but it again shifts the thinking from memory is gone, nothing to do, to a thinking process of memory may be retrievable, which is something I'm saying from the start. So hence the future drugs we can actually bring into, we can innovate and find out that we aim to enhance the engram cell accessibility rather than a synaptic restoration, which might help in targeting the Alzheimer's disease or RTA cases, treat on all such patients. The level of evidences is 11.5. Preclinical animal study again. Grade D strength of recommendation. Now, coming back to the question to the audience. Is the amnesic patient memory gone forever or is it inaccessible? Guys, tell me. What do you think? It's a memory?
Inaccessible, I think.
Yes, exactly. It's inaccessible. And this paper has given a huge turnover and has changed the way of thinking of us all. We have a letter. I have a letter to editor part also over here. I will just add, which might helpful for you guys also. Number one is the points of merit. The landmark study was actually done with multi-method designs and optogenetics is funny we have studied. It's a first cellular proof that memory survives amnesia. It's a robust replication across the brain region with amnesic paradigms. Statistical concerns, no power calculations, and no effect size is actually reported, as I said before. Clinical implication reframes amnesia as retrieval failure, which is directly relevant to our concept, that is Alzheimer's disease therapeutic strategy. Finally, the alternate interpretation, its preserved connectivity may reflect incomplete amnesia rather than a true memory survival. Coming to the future question, yes, always a future research question is needed, right? So here I have few questions for you guys. Number one, what is the molecular mechanism which maintains the engram connectivity without the protein synthesis? And second question I have is, can this non-invasive brain stimulation, any kind of technique we use, the DCS techniques, deep stimulation, replicate the optogenetic engram rescue humans? And third, at what stage of Alzheimer's disease do engram cells die and become merely inaccessible? And also, I have one more question: that does these findings apply to the human retrograde amnesia? Because we did this study in a mice, right? So, is this applicable to the human retrograde amnesia? That's the question I'm putting forward. And the discussion part, in discussion question I'm putting is here: in your clinical experiences, in your hospital setup, like whether it's government or private setup, where it is. Do you think amnesic patients' memories are erased or just locked away? A question that I gave before and a question I'm putting again. So take home message is amnesia is not equal to memory erasure. It's just the engram cells that survives the protein synthesis inhibition, and it is a retrieval failure, not a storage failure. Animal study, it's an animal study only, but compelling concepts, but up to level 5 evidence. Future therapeutic target can be changed, enhancing the engram accessibility, not the synaptic restoration, which may a key role to treat the Alzheimer's memory loss. With that, I'm closing with a quote. Let's change the thinking from memory eraser to memory retrieval. Thank you. These are my references.
So can you stop sharing?
Yes, sir. One.
Are you able to stop the sharing? Okay. So does anyone here have any questions? No questions, is it? So it's time for me to ask questions if you're not going to ask questions. Okay, how many of you are awake? Put your hands up. There's one who is awake, two who is awake, three, four. That's because I slept through this while reading this paper, so I was expecting the same from you as well. Okay, so Manoranjani, you are there, right?
Yes, sir.
Okay, this is the worst ever presentation that I have listened to.
Okay, sir.
The critical appraisal of a paper has to be focused upon, and you have spent very little time on that. Is this the way you present the research paper?
No, sir. Okay, sir. I will take this.
Okay, don't worry, I was just kidding. This is what we call it as fear conditioning. Okay,
Okay, sir. Okay, sir.
you did an extremely good job, way beyond what I could have done. Okay, but
Thank you, sir.
the moment I have actually asked you that, this I wanted to impress upon, because this is what we call as fear conditioning. You might forget everything that has happened in this 1 hour, right? But you wouldn't forget that one sentence that I told you. Even if you liked my initial memory presentation, you would tend to forget it over a month's time. But if I ask you this scolding, if I actually did, but I didn't actually scold you, but if it were true, you wouldn't forget it for even after 30 years. So that's the difference between fear conditioning and one that is without an emotion. Okay, what I'm coming to tell you is that this paper needs certain revisions actually. That's how you need to look at research. They've beautifully done it. The good thing that you need to take from this paper is that they have looked at the NMDA receptor, that's the functional aspect. They have looked at the dendritic spine content, that's the structural aspect. And the third thing is how the function of the mice is. Okay, so is the mice actually shocking, stopping, freezing? That's the functional aspect. So whenever we are looking at the outcome of a study, there has to be a structural assessment, there has to be a function, physiological assessment, and there has to be what does it clinically translate to. So when you address it in three aspects and you prove it on all three planes, that's when you can actually claim the strength of the study. This is one thing that's good about the study. The not so good is they have reported a p-value of less than 0.5, 0.05, but they need to mention what is the p-value exactly to know the strength of association. The smaller, the smallest the p-value, and then you say it's less than 0.05 and that was clinically significant. But you need to report the p-value, which they have not done in this study. And the basic design of the study, let me tell you, is that now Manoranjani will not forget that I have told her about this presentation. That's fear conditioning. Fear conditioning is not the memory that we encounter in our daily life. What we encounter is an episodic memory, and it is not routinely fear conditioned. So, fear conditioned memory will stay for a longer time, easy to be activated. Even if she forgets that I have scolded her right now, if she sees me 50 years later and I tell a hi to her, the first thing that will come to her is this fellow scolded that my presentation is not nice after putting in efforts for 48 hours, day and night. That is easy to get activated. You don't need optogenetics. All she does need to do is see me at some point in her life again. So to activate a fear conditioned memory is easy. And the optogenetics that she has discussed, and the real clinical scenario is RTMS. So the similar to an optogenetics, what we do in clinics is RTMS. Optogenetics is 20 times powered than the RTMS. So do you think RTMS will be actually be able to produce the same effect that this optogenetics has done? Because it's 20 times more powerful than your RTMS. Okay, you can question me if I can improve the RTMS to 20 times more than what it is currently capable of. But what will happen is that RTMS will induce seizures because you're triggering it 20 times more. So there are cons to it when you increase the intensity, when you're really focused, and as long as it's a mice who doesn't question you, you can increase the level of stimulation. Okay, but that is really not possible clinically to increase the stimulation to equivalent of an optogenetics in the mice. So two things that we need to consider from the study while we take it is that one is the strength of stimulation, the fear conditioning to create a memory, and the second is to use optogenetics to retrieve it. These are the two main points that you need to consider in this study. And the final implementation of the final extrapolation clinically as a neurologist, what I would tell you is that when you say there is a memory loss, there is an amnesia, there are two types of memory loss. One is memories that are lost from storage. Point number two is memories that are unable to be retrieved. So there are already existing two types of memory impairments. One where the storage is lost, because when in world there has to be, if there is a storage process, there is a retrieval process, then there has to be a problem which affects the storage, there has to be a problem which affects the retrieval. So this paper summarizes that memory is all about problem with the retrieval, which is not true because there are two types of problems. One is a storage problem and other is a retrieval problem. So the retrieval problem is that when you are when you have the storage but you are unable to retrieve it. The other thing is that are when you are able to retrieve but you don't have the storage, or when there is no storage, there is no question of retrieving it. So this is the classical cortical dementia, that you are losing the storage. And the second part, you are unable to retrieve it, is the subcortical dementia. So I'm not going to go further into it. Both are available, both are existential. There is a disease which is a problem of storage, there is a disease which is a problem of retrieval. And the problem of storage is the classical example is the Alzheimer's dementia. The classical example of inability to retrieve things is vascular dementia. So the way we treat these two are different. The second one, where there is a problem of retrieving, we give them cues to improve. Let me tell them name of a bird, and we later give them three choices and ask them to find out which was the bird that we had told earlier. So that is giving them cues. It works to some extent like optogenetics, where it triggers the pathway to get the information, but not to the extent amplitude or the magnitude to which the optogenetics actually work. So that's the subcortical dementia. So there are a whole lot of causes for subcortical dementia. There is a whole lot of causes for cortical dementia. And the way we treat cortical dementias are different than the way we treat subcortical dementia. So this is the final take home point. And last two things is that honestly, Manoranjani, I did not expect this level of presentation from an undergraduate, because this is the area where even DM residents struggle a lot with regards to the engrams of memory, and you did an amazing job. And I really enjoyed your presentation. And my sincere advice is take a clinical topic next time. That would be easy for me to read through, and I wouldn't sleep through the article while I read a mouse model. Sometimes I realize that I am a veterinarian when I read this paper. It was a wonderful presentation. So, anyone has any questions here? Any questions? If not, I'll hand over it to Kirtan. Was this session useful? Were you all able to understand the memory and what happens when you lose memory, the research aspect? How do you frame a question? That's beautifully done. I don't have any more points to add to what she has done with regards to the research aspect, and that was amazing. You can go through her presentation over and over again, and you will understand it much, much better.
Yes, sir. It was very clear, sir. We understood a lot, sir.
Okay, thank you so much. It was wonderful being with young minds today. I thank the opportunity for being here. Now I hand over to Kirtan.
Yes. Thank you, Manoranjani, for that electrifying and insightful, well presented session. As we come to the end of this enriching session, it is time to express our heartfelt gratitude to Dr. Guhan Ramamurthy, sir, for his wonderful insights. And we have learned a lot from your session, sir. On behalf of RIT 2026 and IMA-MSN TNSB, I extend my sincere thanks to our presenter, Ms. Manoranjani. And then I would also like to thank our organizing team, Mr. Arunagiri Gunasekar, convener IMA TSN TNSB, Dr. Praveen Selvanandam, Research Committee Head, IMA JDN TNSB, and our dedicated tech team members, Akash Harikrishnan, Purvik Narayan M, Niveda S, and Jeevana J. Finally, thank you to all the participants for your active presence and enthusiasm. We hope this session has added value to your academic journey and sparked your interest in research. With this, we conclude today's session. Thank you all once again. Have a great evening ahead. Actually, good night.
Thank you. Sir, thanks for your time, sir.
Thank you so much, sir.
Very insightful.
Thank you,
Thank you,
sir.
sir.
Thank you very much.
Manoranji, thanks for your efforts. It was very good.
Thank you, sir. Thank you, sir. Thank you all for joining.
The meeting is 10 days. You can leave. Thanks for attending.